Let me start with you, Dr. Malone. I think it was in...
And we can make it Robert, it's okay.
Lovely, I love that. So I think it was in 1987 that you performed a landmark experiment where you were mixing strands of messenger RNA droplets with fat to create the molecular, what people call a stew or something. But that all sounds very technical. Have I got that right? Can you give us a little idea of the kind of provenance of that, if you would?
So you're asking what is the genesis of the original mRNA inventions?
Exactly.
I was working as a graduate student in a postdoc lab at the Salk Institute in the molecular biology and virology labs, passionately pursuing a career in gene therapy.
Right.
I wanted to be a MD that would cure pediatric genetic disease. That was my dream. And the way to do it at the time, the leading technology involved retroviruses. And San Diego was the place to be. The two leading people were there, Ted Friedman, who'd originally come up with the idea in the 70s, and Inder Verma, who had come from David Baltimore's lab and was really one of the world's experts in retroviruses, which is the main platform that was being used. And so I weaseled my way in as a graduate student into an intensely competitive postdoc lab at the Salk, and had to carve out some niche where I wasn't going to get just overrun by the postdocs. And so that niche was how did the RNA assemble into a retrovirus particle? And I thought it was going to be simple. I could just make RNA and it would be genetically modified, have the things that I wanted, and then I could use the same technology that was used for putting DNA into cells, in cultured cells. I could use it for RNA, and everything would be hunky-dory, and I could do reverse genetics and cool science and get along with that. None of that turned out to be true. Nobody had really made large quantities of pure mRNA that could be used to produce protein in cells. And all the tech that existed at the time for putting DNA, this double-stranded version, into cells just didn't work. And I tried them all, and I spent quite a bit of time developing the recombinant DNA system and how to cook the RNA in large quantities. It was really scary. It was like $5,000 worth of enzymes in a single tube. For a graduate student at age 27 in the 1980s, that was high risk experiment. I got those things solved and still could not get the RNA into cells with liposomes. I went to UCSF, worked with Judy White, one of the top liposomologists at the time. Nothing seemed to work very well. It's very frustrating. And then Tony Hunter, a brilliant scientist at the Salk and kind of occasionally mentioned for a Nobel, came to me and he said, "Hey, Robert, I heard about this new tech out of this company, Syntex, involving positively charged fats, and I think you ought to look into it." And so I made the phone call, set up the arrangements, and as a good graduate student, well-trained, I wrote initial patent disclosures, invention disclosures, and all that before we did that. And they couldn't, at Syntex, they couldn't get RNA to work for the reasons that I'd been banging my head against the wall for a couple of years, because they didn't know how to make it and all that other infrastructure stuff. So they had the positively charged fat tech at Syntex, and I had all of the manufacturing pure RNA and knowing how to detect it and all that tech. And they shipped me their reagent, and it was like, lightning. Suddenly everything worked. And there was not a cell line at the Salk Institute that I could find that I couldn't put RNA into and get luciferase protein. This was the first real use of luciferase in this way also. This is this firefly protein that was actually used by Pfizer in their studies to justify these current jabs that are going forward, instead of actually using the protein that was the spike. So that's how that all came together. And then having done that, I was helping teach an embryology course, and I had access to a variety of different types of embryos, like frog embryos and chick embryos. And I just kind of a lark, I said, well, let's see what happens if we take this and put it on that. Lo and behold, that worked. It's like nothing went wrong. Everything worked. It was amazing time. And then the fight started over the intellectual property.